etc. Now let's move into our presentation, starting with best practices for abbreviated new drug applications in GDUFA III. Our presenter in this presentation, our first presenter, is Commander Chitra Mahavathan, is the Director of the Division of Bioequivalence Process Management and the Office of Bioequivalence in OGD. She leads a division of regulatory health project managers who are responsible for managing all the processes associated with bioequivalence reviews throughout the drug product life cycle for ANDAs. Our second presenter in this presentation is Captain Craig Keister. Captain Keister is a division director in the Office of Product Quality in Cedar. He manages the project management of staff responsible for co-leading the review of quality portion of ANDAs. In 2001, he started his career with FDA's OGD as a chemistry project manager. Within OGD, he's also held positions as a regulatory review officer and a microbiology project manager. Our second presentation is on the submission of In-Vitro released test data and information for topical drug products under ANDAs. Our presenter, Dr. Wei Zhang, is an assessor in the Office of Bioequivalence in OGD. She has experience in bioequivalence assessment for drug products across a wide range of therapeutic areas with different dosage forms. Hui serves as one of the division representatives in the Topical Product Specialty Focus Group. Our third presentation is on the submission of in-vitro permeation test data and information for topical drug products under ANDAs. Our presenter, Dr. Archana Manirekar, serves as a bioequivalence assessor in the Office of Bioequivalence within OGD. In her current role at OGD, she's responsible for collaborative assessment of ANDAs and controlled correspondences. She's also responsible for providing OB assessments of pre-ANDA submissions, including novel bioequivalence approaches for topical products, as well as industry outreach aimed at enhancing the quality of ANDA submissions for topical products. Next, we present on considerations for alternative bioequivalence in vitro study information submitted in nasal drugs. Our presenter, Dr. Vipra Kundur, is a pharmacologist in the Division of Bioequivalence 1 within OGD. She's responsible for assessing the bioequivalence of various dosage forms of generic drugs. She's also involved in addressing controlled correspondences and pre-ANDA meeting packages. She is a focal point expert for nasal and inhalation drug products within the division and conducts secondary reviews for these complex dosage forms. She's also actively involved in the review panel for product-specific guidances for nasal and inhalation drug products. Our fifth presentation is on Structured Submission and Review, Module 3. Our first presenter in this presentation is Dr. Scott Gordon. Dr. Gordon's been responsible for a range of activities to standardize data, including real-world data derived from health information technology for use in clinical research and pharmacovigilance, as well as standardization of pharmaceutical quality and manufacturing data for submission to FDA. Our second presenter in this presentation is Dr. Norman Schmuff. Dr. Schmuff joined FDA as a chemistry reviewer in 1990. Subsequently at FDA, he's been a team leader, branch chief, and associate director. He's participated in 4 ICH expert working groups, including what was then the M2 ECTD group. Currently, he's the technical lead for the PQ-CMC project. Our final presentation before the Q&A panel is on information to include with cover letters. Our presenter, Lieutenant Commander Nemi Matthews, is a regulatory project manager within the Division of Project Management and the Office of Regulatory Operations within OGD. She joined FDA in 2014 and has held positions within OGD as well as the Office of Regulatory Affairs. In addition to her current regulatory project management activities, Nemi also serves as a co-lead in GDUFA III implementation work group for Facility Not Ready pathway. Please join me in welcoming our first presenter, Commander Chitra Mahavathan.
Hello, my name is Commander Chitra Mahadevan, and I am the Director of the Division of Bioequivalence Process Management in the OGD Office of Bioequivalence. Thank you for the opportunity to speak today on best practices for abbreviated new drug applications in GDUFA III, a bioequivalence perspective. The objectives for today's presentation are to explain how to engage with the Office of Bioequivalence during various stages of the ANDA lifecycle, to provide an overview of bioequivalence communications during ANDA assessment cycles, and to review best practices. So first, I will touch upon ways to engage with the Office of Bioequivalence, or OB, in GDUFA III during various stages of the ANDA lifecycle through both meetings and written communication. So prior to ANDA submission, there are several pre-ANDA meetings that can be requested. For product development meetings, there has been no change from GDUFA II, and in general, these meetings provide an opportunity to discuss specific scientific issues related to a complex product, such as a proposed study design or an alternative approach. Pre-submission meetings have been expanded a bit in GDUFA III to provide a prospective applicant the opportunity to present unique or novel information that will be included in the ANDA submission. During these meetings, which typically occur approximately 6 to 8 months before ANDA submission, you can expect to engage with those from FDA who attended the product development meeting for the same product if one was held, as well as ANDA discipline assessment team members, including the OB team, that may assess the ANDA upon receipt. New meetings in GDUFA III include pre-submission product-specific guidance teleconferences and pre-submission product-specific guidance meetings. When a new or revised PSG is published and a prospective applicant or applicant has already started an in vivo bioequivalence study, a PSG teleconference may be requested to obtain feedback on the potential impact the new or revised PSG may have on the development program. If further feedback is required after a PSG teleconference, a controlled correspondence or a PSG meeting request may be submitted. And the purpose of the PSG meeting is to discuss the scientific rationale for an approach other than that recommended in the PSG. Prior to ANDA submission, OB will utilize written communication to respond to controlled correspondence. OB typically assesses and responds to inquiries related to inactive ingredients, specific drug development questions related to bioequivalence, risk evaluation and mitigation strategies with elements to assure safe use, or REMS with ETASU protocols, and when a prospective applicant requires further feedback after a pre-submission PSG teleconference. During the ANDA assessment, new in GDUFA III, you can request a post-submission PSG teleconference or post-submission PSG meeting. You can also request a mid-cycle review meeting, which is the same in GDUFA III, except for the revised name. In GDUFA II, it was called the Mid-Review Cycle Meeting, or MRCM, and now in GDUFA III, it is called the Mid-Cycle Review Meeting, or MCRM. And the purpose of the MCRM is for the applicant to ask for rationale for any deficiency listed in the Mid-Cycle Discipline Review Letter, or DRL, or to ask questions related to FDA's assessment of ANDA data or information. New data or information is not accepted for this type of meeting. An MCRM may be requested for complex products with a granted product development meeting, some eligible complex products with a pre-submission meeting only, and for eligible ANDAs designated as competitive generic therapy. In addition, you can also request an enhanced mid-cycle review meeting, or an EMCRM, and this is a new meeting type in GDUFA III. Complex generic products with a granted product development meeting and some with a granted pre-submission meeting only are eligible for an EMCRM. And the purpose of this type of meeting is to provide an ANDA applicant with the opportunity to ask questions related to a proposed scientific path to address possible deficiencies that have been identified in the mid-cycle DRL. An applicant may ask questions about potential new data or information, which FDA will discuss at the meeting. However, FDA will not provide substantive assessment at this meeting. The goal date will be extended by 60 days, and the DRL response due date will be recalculated from the EMCRM date. So for instance, if the initial DRL response due date was 30 days, now the response will be due 30 days after the date of the EMCRM. And as far as written communication, new in GDUFA III, during the ANDA assessment cycle, a control correspondence may be submitted if seeking further feedback after a post-submission PSG teleconference or to seek a covered product authorization. All other correspondence during an ANDA assessment cycle will be general correspondence. So there are several ways to engage with OB post-complete response letter or post-CRL. So an applicant can request a post-CRL clarification teleconference, which allows an applicant to seek clarification on CRL deficiencies. Non-clarification questions will not be answered. And new in GDUFA III is a post-CRL scientific meeting. The purpose of this meeting is to provide an applicant scientific advice on possible approaches to address CRL deficiencies that are related to establishing equivalence. Applicants can request this type of meeting even if there was no product development meeting. And for the post-CRL scientific meeting, questions related to both establishing equivalence as well as clarification questions may be submitted. You can also submit a post-submission PSG teleconference and post-submission PSG meeting. In addition, new in GDUFA III, a control correspondence can be submitted post-CRL. Post-approval in GDUFA III, you can now engage with OB by submitting a control correspondence. Some examples of post-approval questions we see in OB include inquiries about reference-listed drug availability and post-approval bioequivalence study recommendations. So now I will provide an overview of bioequivalence communications during ANDA assessment cycles in GDUFA III. The ANDA assessment program enhancements in GDUFA III are aimed at improving predictability and transparency, promoting efficiency and effectiveness of the review process, minimizing the number of assessment cycles needed for approval, increasing the overall rate of approval, and facilitating greater access to generic products. An information request, or an IR, is sent during the assessment cycle and is used to request further information or clarification that's needed to complete the discipline's assessment. The issuance of an IR does not stop the assessment clock. And deficiencies in IRs will be identified as major or minor, which is new in GDUFA III, and generally, these will include a response due date. Bioequivalence IRs are typically communicated by email and will come from the OB project manager. Discipline review letters, or DRLs, convey preliminary thoughts on possible deficiencies at the conclusion of a discipline's assessment. First assessment cycle DRLs will typically be issued by the midpoint and will include a response due date. And this is new in GDUFA III. Also new in GDUFA III, DRL deficiencies will be classified as major or minor. If minor DRLs are responded to by the requested due date, that response will be reviewed within the original goal date. However, for responses to major DRLs and responses to minor deficiencies which require comparable resources as major deficiencies, such as a consult, these will be classified as major amendments and the goal date will be extended. And this is to minimize the number of assessment cycles that are needed for approval. We will also issue IRs and DRLs after the midpoint in cases where one or more deficiency can be resolved through an IR or DRL, and as a result, the ANDA may be approved or tentatively approved in that current cycle. For these IRs and DRLs, major and minor deficiencies will be identified, and a due date may be assigned. The goal date may be extended by 90 days if the response is received by the due date, addresses all identified issues, and does not contain unsolicited information. And note that DRLs without a response due date may signify a forthcoming complete response letter. So now I will discuss bioequivalence best practices. For control correspondence, please submit through the CDER Direct NextGen Collaboration portal and not to individuals at FDA. Also, ensure that your inquiries are specific and contain sufficient detail. Please be sure to limit requests to one discipline to facilitate timely assessment and response, and please do not submit the same questions utilizing multiple submission routes. For inactive ingredient requests, please submit no more than 3 inactive ingredients and no more than 3 proposed levels for a drug product. So this means you can, for instance, submit 3 inactive ingredients with 1 level each, or 1 inactive ingredient with 3 proposed levels. Also, only submit inactive ingredients to be evaluated and their proposed levels. If they're not to be evaluated, please do not submit them. And please be sure to identify the reference listed drug, including the specific drug product strengths. For cover letters, please submit an outline of impacted disciplines, information on any previous communications you've had with the agency, such as related control correspondence or pre-ANDA meetings. For control correspondence cover letters, please indicate whether the inquiry is related to an ANDA. And for ANDA submissions, please indicate in the cover letter if there is an active pharmaceutical ingredient source change or submission of new bioequivalence studies conducted at a new contract research organization. For IRs and DRLs, please respond completely and by the response due date. Partial responses will not be accepted, and if responses are not received by the due date, the deficiencies may be included in the complete response letter and assessed during the next assessment cycle. Also, please be clear and concise and only submit the requested information, as unsolicited information may impact the assessment timeline and extend goal dates. If you require additional time to respond to an IR or DRL, please submit an extension request as soon as possible. And the point of contact for bioequivalence IRs and DRLs is the OB project manager listed on the letter. Extension requests will be reviewed on a case-by-case basis. So this slide lists the FDA contacts. For general ANDA questions, please reach out to the OGD RPM. For questions related to bioequivalence IRs and DRLs, please reach out to the OBPM. And for any quality-related questions, please reach out to the OPQ RBPM. So now I will go through 2 challenge questions. So for question number 1, in GDUFA III, during the ANDA assessment cycle, a control correspondence may be submitted in which of the following situations? A, after a PSG teleconference. B, when seeking a covered product authorization. C, for any clarification questions. D, A and B only. Or E, all of the above. Please take a few moments to think about the correct answer. And the correct answer is D, A and B only. So a control correspondence may be submitted during the ANDA assessment cycle if one is seeking further clarification after a PSG teleconference or when seeking a covered product authorization. Other correspondence will be considered a general correspondence. So for challenge question number 2, which of the following is true about first assessment cycle DRLs in GDUFA III? A will be issued by the assessment cycle midpoint. B, deficiencies will be classified as major or minor. C, a response due date will be included. D, responses to major DRLs will extend the goal date. Or E, all of the above. Please take a few moments to think about the correct answer. And the correct answer is E, all of the above. So first assessment cycle DRLs will be issued by the assessment cycle midpoint. They will be classified as major or minor and include a response due date. In addition, responses to major DRLs will be classified as major amendments and the goal date will be extended. So the next 2 slides include various helpful resources for you to refer to. So in conclusion, please be familiar with the expanded definitions of controlled correspondence, new meeting types, and communication enhancements in GDUFA III. In addition, follow the best practices discussed to reduce the number of bioequivalence assessment cycles. For any questions related to bioequivalence IRs and DRLs, please reach out to the OB project manager. And thank you for your time and attention today. I look forward to answering any questions during the panel session.
Good morning. My name is Captain Craig Keister. I'm a Division Director in OPQ's Office of Program and Regulatory Operations. Today I'm going to explain how best to engage with OPQ during the ANDA life cycle, how to provide an overview of OPQ communications during and after assessment cycles, and finally, talk about some best practices and common themes we see on our end. Hopefully you'll find this information helpful. First and foremost, the RBPM is the primary point of contact for all OPQ-related inquiries. The RBPM and OPRO, and they work closely with the RPMs in OGD, as well as the assessment team in OPQ on all of your applications. Additionally, they issue all communications related to quality during the assessment cycle. If for some reason your RBPM is unresponsive, please be mindful of the layers in the organization to elevate to, starting with their branch chiefs, division directors, deputy office director, or office director, prior to reaching out to more senior FDA leadership. Next up, I'd like to talk a little bit about the information request letter, or the IR letter. It's very common and very valuable means of communication between us and industry. It's a tool that allows us to solicit for additional information or clarification that we need in order to complete the assessment cycle. The IR could be single discipline or multidiscipline. When issuing the IRs, the RBPMs will typically request a response date, and you will receive a communication via either phone, email, or letters. Next up is the discipline review letter, the DRL. That is more comprehensive than the IR. It's the assessment team's preliminary findings, and it's issued when our assessment has been substantially completed. This is usually around the midpoint of the assessment cycle. Like the IR, you will typically be asked to respond by a certain date. Responding to IRs and DRLs. Extension requests should be made as soon as possible by contacting the RBPM. The RBPM and assessment team will work together to determine if an extension will be granted. Your extension request should be done in a manner that provides you enough time to completely respond to the IR DRL, and we will frequently not allow multiple extensions. I would also like to point out partial responses will not be accepted, and the timing or unsolicited information in either of these submissions could potentially extend the goal date of the application. Next up, we'll talk about OPQ communications, and particularly the meetings. First up would be the mid-cycle review meeting. It's for the applicant to ask for rationale for any deficiency identified in the mid-cycle DRL, and or to ask questions related to the FDA's assessment of data or information in the ANDA. An applicant may not present any new data or information at this meeting. A mid-cycle review meeting will take place within 30 days after the date the sponsor submits a meeting request. A new meeting we have is the enhanced mid-cycle review meeting. The purpose for this meeting is for the applicant to ask questions related to proposed scientific path to address possible deficiencies identified in the mid-cycle DRL. An applicant may ask questions about potential new data or information to address any possible deficiencies identified in the mid-cycle DRL. FDA will discuss the data and information, but will not provide substantive assessment of data or information provided by the applicant at that meeting. If an enhanced mid-cycle review meeting is requested, the meeting will take place within 90 days after issuance of the last mid-cycle DRL. FDA will extend the ANDA goal date by 60 days if an enhanced mid-cycle review meeting is requested. It will also extend the response due date for any relevant DRLs by recalculating the response date starting from the date of the meeting. For example, if we initially requested 30 days after the DRL, it will now be due 30 days after the enhanced mid-cycle review meeting. An applicant may submit an unsolicited amendment after an enhanced mid-cycle review meeting, which could result in additional goal date extensions as well. Post-CRL teleconference. That's used to seek clarification concerning deficiencies identified in a CRL. FDA will grant appropriate requests for teleconferences requested by applicants upon receiving first cycle CRLs and upon receiving subsequent CRLs. An appropriate request is one that clearly identifies the specific deficiencies to be discussed and the reason why such deficiencies are not clear. FDA will provide a scheduled date for 90% of the post-CRL teleconferences within 14 days of the request and conduct 90% of the teleconferences, if granted, within 30 days of the written request. Controlled correspondences. So under GDUFA II, OPQ received approximately 700 controlled correspondences annually. So approximately 2,800 of those in the previous 4 years. Common themes of those were stability data and how much was needed, container closure, changes, bracketing of batches, microbiology, batch size, packaging changes, and questions. The control correspondence on the amount of stability data increased dramatically after the publication of FDA guidance, development of ANDAs during the COVID-19 pandemic, questions and answers. Under the G2 framework, correspondence seeking regulatory or scientific advice after issuance of the CR, TA, or approval letter was considered general correspondence. Under the new G3 regulations, these are now submitted as controlled correspondence. Applicants now have the option to submit controlled correspondence to seek clarification. In OPQ, we perform an IQA with a multidiscipline team and the PMs on each application. This team, in most cases, will be assigned to address the controlled correspondence. So, to sum up, during the ANDA assessment cycle, it can be submitted if an applicant needs feedback following a product-specific guidance teleconference or to seek a covered product authorization. If it's after the ANDA assessment cycle, so following a CR, TR, or full approval, it'll be submitted as controlled correspondence. DMF unsolicited amendment issues. Poorly timed unsolicited amendments continue to adversely impact the application timelines. This can result in goal date extensions, unnecessary CR letters to the DMF and or the applicant. Frequently, unsolicited amendments are not necessary to be submitted at the time they are. And communication between the applicant and DMF holder regarding DMF submissions is the best solution. So some tips to avoid unsolicited amendments. DMF holders and applicants should share the information in a timely way. Any plan changes to the DMF, as well as action dates and priorities for the referencing applications. If it is not clear whether an unsolicited DMF will impact an application timeline, do the following. Reach out to your customers to get information on their application timelines. Reach out to DMF OGD and see if we can provide helpful guidance on the submission timing. And most importantly, understand the consequence of a poorly timed DMF submission to your customer's ANDA. It can likely result in goal date extensions depending on the date that it's received, and the ANDA goal date will be extended from the date of the DMF amendment. So if that amendment is submitted very late in an ANDA review cycle, they potentially could have a long extension to the ANDA. The length of that extension is determined by the nature of the amendment. If it's a major or a minor, it could result in them losing an exclusivity or a forfeiture date, and withdrawal of an amendment will not undo an already extended goal date. So some GDUFA III DMF enhancements. So first up is a DMF prior assessment. A DMF holder may submit a request for the assessment of an original DMF previously unreviewed 6 months prior to the planned submission date of an ANDA. And that's for an original, an amendment, or a PAS. FDA will grant and review the DMF prior to ANDA submission when the planned application submission meets certain criteria. Some of that criteria: all patents and exclusivities will expire within 12 months of the planned submission date. There are fewer than 4 approved therapeutically equivalent drug products, including RLD in the Orange Book. No blocking patents or unexpired exclusivities in the Orange Book. And the applicant is not seeking to carve out any conditions of use. In addition, submission for a drug product that could help mitigate or resolve a drug shortage and prevent future shortages, or if it's related to a public health emergency. So FDA assessment of solicited DMF amendments. FDA will assess certain solicited DMF amendments related to original ANDAs and PASs upon receipt, even if the original ANDA or PAS in which the DMF is referenced is not currently under assessment. Priority is given to those amendments related to ANDAs for which acceptability of the DMF assessment may result in a full approval on the next application cycle. Assessment comments issued to DMF holder at least in parallel with issuance of the review comments relating to the DMF that ANDA references.
Now let's discuss some common deficiencies in the DMFs. We'll start off with non-mutagenic organic impurity controls. All potential impurities need to be included in the discussion. This includes starting materials, intermediates, including in situ intermediates as appropriate, byproducts, and impurities in the starting material and intermediates, as well as their continued reactions in the process.
Common deficiencies in the DMF. Mutagenic impurity controls. Failure to use QSAR methodologies that are ICH M7 compliant in the hazard assessment. Insufficient data is provided when upstream controls are established at higher limits. Incorrect threshold of toxicological concerns is proposed. Failure to limit the total mutagenic impurities when multiple impurities are present, and failure to show the analytical method is suitable for its intended use. Drug product considerations. Extractable leachable studies for key packaging components. Utilize correct safety concern threshold for analytical evaluation thresholds. Select study conditions pertinent to specific attributes of the product. Maximum daily dose for impurity and extractable leachable qualifications. Impurity qualifications besides the PharmTox qualification for small molecule APIs, and any device differences between generic and RLDs. Facility assessment, what we need. Please ensure that all current facilities, including all relevant DMF facilities and their responsibilities, are clearly listed on the Form 356H. Process development and bridge to commercial scale. Commercial process descriptions, process flow diagrams, and sterilization validation packages for sterile products. We would also like to note that you are now required to verify that all facilities are ready for inspection. Common manufacturing deficiencies. New batches required due to significant failures with submitted batches with no root cause of understanding provided. An incomplete facility responsibility listing on a 356H. And process parameters and in-process controls not supported by process development knowledge in the application. The cover letter. We have attached is a hyperlink for the cover letter attachments for control correspondence and ANDA submissions. We ask that you use this when identifying to identify ask you use this when preparing submission cover letters. Be sure to identify all applicable disciplines for which you're submitting information, and that's for the amendments in particularly. We'd also like to remind you that secure email is required or strongly recommended. If you don't have this, you can easily request it by sending an email to secure@fda.hhs.gov. The templates and the guidance appendices will make for more efficient triage process. I'm not going to read all these off, but the next 3 slides provide links to resources that will be helpful for you for managing your applications. CEDAR guidance, GDUFA webpage, good ANDA practices, cover letter, attachments for control correspondences, ANDA submissions, and on. So in summary, please reach out to all RBPMs for all quality-related communications. Completely respond to all quality IR DRLs by the requested response date, and be familiar with your ANDA assessment timeline and all possible communications. Actively and frequently communicate with your DMF holder. And lastly, take advantage of the many new ways to communicate with the agency with the GDUFA III enhancements. Thank you very much for your time. Be familiar with your ANDA assessment timeline and all possible.
I'm from the Office of Bioequivalence in the Office of Generic Drugs. In this presentation, I will speak on the submission of in-vitro release test data for topical drug products under Abbreviated New Drug Application. This presentation reflects the views of the author and should not be construed to represent FDA's views or policies. After this presentation, the audience should be able to identify the key components that constitute a high-quality IVRT submission, describe the level of detail expected for each key component, and know the appropriate module for each component to be submitted in the electronic common technical document. I will start off with a brief introduction on the in-vitro release test, and then explain the agency's expectation for a high-quality IVRT submission. In the next few minutes, I will share with you some submission-related common deficiencies. FDA published the draft guidance for industry, in-vitro release test studies for topical drug products submitted in ANDAs in October 2022. In recent years, there has been an increasing trend of topical drug product applications that contain in vitro studies, in place of the traditional comparative clinical endpoint study to support demonstration of bioequivalence. The characterization-based approach includes in vitro release test, in vitro permeation test, and physicochemical and structural property studies. IVRT is utilized to assess the rate of drug release from a proposed generic topical product, comparing to its reference standard for the purpose of supporting a demonstration of bioequivalence. Please note, the topical products here may include gel, emulsion, lotion, cream, ointment, or other semi-solid or liquid-based dosage forms applied to the skin, but do not include the transdermal delivery system, such as patches or fumes. To develop an IVRT method and eventually use it to support a demonstration of bioequivalence, the process includes 3 phases: method development, method validation, and pivotal comparative IVRT study. In general, there are 4 major components. Usually, the applicants prepare a separate study report for each of the major components. Those study reports include stepwise details of the test procedures that enable the assessors to reconstruct in their mind how the studies were conducted. Also, in the submission, all the raw data are included so the agency may verify the study results. In the next few slides, we will discuss the agency's expectation with regard to a high-quality IVRT submission. Like other in vivo bioequivalence studies, The summary tables for each component are to be submitted in Module 2.71. Detailed study reports, protocols, relevant standard operating procedures, and other supporting documents are to be submitted in Module 5.312, except for anything pertaining to the sample analysis, the analytical method validation, and the documentations related to the study sample analysis, including the chromatograms, raw data printout, should be submitted in Module 5.314 in the respective subfolders. In this slide, we will focus on the IVRT method development. Summary information relevant to IVRT method development, including but not limited to the following information, may be submitted in tabular format in Module 2.71. Given that multiple conditions may be tested to optimize your IVRT method, it is recommended to list all the conditions tested for each parameter. A detailed IVRT method development report containing the above information as well as supporting documents should be submitted in Module 5.312. One of the common deficiencies is that applicants described all the parameters they had tested, but did not include any rationale for the selection of parameters. Another deficiency is that missing study details in the report. For example, the stepwise procedure for dose application. Please include information such as the tool you use in the dose application, whether the dosing chamber is occluded or not, is dosing staggered or sampling synchronized, or vice versa. Now let's move on to the method validation. Similar to the method development, in Module 2.71, summary table of IVRT method validation should include the general information and a method description. A report of IVRT method validation should be submitted in Module 5.312. Please refer to the IVRT guidance for the definitions of validation parameters. It is commonly seen an IVRT submission miss one of the following information, such as apparatus qualification data, receptor solution sampling qualification data, a detailed procedure for dose application, or a component and conversation table of the altered formulation, or room temperature and relative humidity logs. Just like a regular submission for in vivo bioequivalence study, you need to submit a summary table for analytical method validation in Module 2.71 and a standalone report for analytical method validation along with SOP, raw data, and the representative chromatograms in Module 5.314. I want to emphasize here, please do not forget the stability data under assay condition. Now we move on to the last key component, pivotal IVRT study. The contents in the summary tables are very similar to that in the method validation. You provide the final test parameters of the pivotal study design, as well as the general information such as study number and the test site. Please note, as a pivotal BE study, the test site is subject to FDA's inspection. In the report of pivotal IVRT study, please make sure you include the test and the reference product information, such as the batch size, the manufacturing date of the test product, and expiration date for the reference product, and the potency for both batches. The requirement for submission of sample analysis data is the same as other in vivo PKBE studies. That should include 100% of the raw data and 20% of the chromatograms. Please submit all the rejected, re-injected, and repeat analysis. The individual concentration data should be submitted in the appropriate format. To summarize, the agency often identifies missing or incomplete information in the original submission. Many of the common deficiencies described today can be avoided. Please submit all necessary information or data that enable the reconstruction of study process and the verification of results. Now it's time for the challenge question. What are the key components of an IVRT study submission? The answer is all of the above. That concludes my presentation. I would like to express my gratitude to my colleagues and supervisors for their invaluable assistance in preparing for this presentation. Now I hand it over to the next speaker.
Good morning. It's great to be here today to talk about IVPT submissions. As we know, IVPT studies may be used to characterize and compare the rate and extent of a drug's bioavailability at or near a site of action in the skin from a test topical product and the reference standard.
To talk about IVPT
Thus,
submissions.
IVPT studies are important components of the characterization-based bioequivalence or BE approach for generic drugs as outlined within product-specific guidances of several topical products. So let's jump right in and see if we can demystify high-quality submissions for IVPT studies. This presentation reflects my views and should not be construed to represent FDA's views or policies. Our learning objectives are to identify key components of a high-quality IVPT submission, understand the key submission expectations for each key component, and clarify the organization of IVPT information and data within the ANDA submission. The 5 key components include relevant to the IVPT methodology, development and optimization, referred to as IVPT method development, validation, including sensitivity and pilot studies, referred to as IVPT method validation, and IVPT pivotal evaluation based on the validated methodology. Relevant to the analytical methodology, validation of the receptor sample analysis methodology, also referred to as analytical method validation, and IVPT method validation and pivotal study sample analyses. Now, let's look at some key submission expectations for all IVPT studies, as well as a few that are specific to each of the 5 key components. Key submission expectations relevant to all IVPT studies include submissions of details relevant to each IVPT study, such as study objective, method parameters, study procedures, operators involved, and conclusion. For examples of IVPT method parameters and study procedures, please refer to Appendix B provided in the PDF of these slides. Other key expectations for all IVPT studies include the submissions of details that are relevant to each experimental IVPT run, such as diffusion cells run in parallel, identified by unique cell IDs, cells that were dosed, relevant product lot numbers, and experimental run dates. Details relevant to each product lot, such as lot number, product strength, formulation, batch size as applicable, as well as assay potency with container content uniformity if available, manufacturing or expiration dates as applicable, and for any altered formulations, rationale for the alteration made, and description of the altered manufacturing process. Details relevant to each skin section, such as donor ID, age, race and sex, supplier information, anatomical site of skin collection, and individual measurements mean and percent CV for skin thickness and barrier integrity test results. Details relevant to the handling of each skin section, such as conditions at which skin was received and subsequently stored, date and time of each retrieval from and restorage in the deep freezer, and the number of freeze-thaw cycles to which the skin was subjected. Lastly, submission of details of any protocol or SOP deviations or experimental observations that may influence the interpretation of the study results. Moving on, key expectations for IVPT method development studies include submissions of recommended details as relevant to all IVPT studies, documentation of all systematic efforts leading to the selection of a IVPT methodology suitable for demonstration of BE, complete details of the optimized IVPT methodology comprised of IVPT equipment and method parameters, acceptance criteria and study conditions, along with well-controlled study procedures documented in detailed SOPs. Another key expectation is the submission of spreadsheets containing raw concentration and PK data for method development studies showing all relevant calculations. For the preferred data format, please see Appendix A in the PDF of these slides. Lastly, it is expected that IVPT methodology that is first thoroughly optimized, then adequately validated and utilized in the pilot study is used unchanged in the subsequent pivotal study. Key expectations for IVPT method validation studies include submission of adequate information and data to support specific qualifications and controls that are related to the suitability of IVPT equipment, suitability of skin sections selected for dosing, suitability of the receptor solution, suitability of the sampling procedure, characterization of the permeation profile and range, characterization of the precision and reproducibility of results, characterization of depletion of the applied target dose over the study duration, validation of the method selectivity, consistency of ambient study conditions, suitability of the applied target dose amount and dose duration, and lastly, submission of study results that are supportive of method robustness, if available. Other key expectations include submissions of recommended details that are relevant across all IVPT studies, detailed protocols and SOPs, adequate evidence to support control and consistency of study procedures, conditions, and method parameters, as well as submission of spreadsheets containing raw concentration and PK data for all validation studies showing relevant calculations. Moving on, key expectations for IVPT pivotal study include submissions of, again, recommended details that are relevant to all IVPT studies, relevant protocols and SOPs, details of randomization, blinding, and unblinding procedures, adequate evidence that supports control and consistency of study procedures, conditions, and method parameters, as well as SAS transport raw concentration PK datasets for the pivotal study. Next up, key expectations for analytical method validation studies include submissions of complete details of the sample analysis method and experimental procedures, relevant protocols and SOPs, submission of complete details of each study involved in the analytical method validation such as linearity, recovery, etc., along with adequate assessments of drug stability and dilution integrity as applicable. Last but not least, key expectations for analyses of IVPT method validation and pivotal study samples include submissions of relevant protocols and SOPs, 100% numerical raw data for all analytical runs, complete details of each analytical run or sample, rejection, reinjection, repetition, or reintegration as applicable, and submission of adequate representative chromatograms. Lastly, relevant to each analytical run, submission of details such as run ID, analyte name, dates of extraction and assay, donor ID, instrument ID, analytical run status such as accepted, rejected, re-injected, etc., and a brief run description. Now let's see how these IVPT data and information should be appropriately organized within the ANDA submission. Information and data relevant to IVPT studies, such as method development, validation, and pivotal studies, belong in Module 5312. Thus, study reports, protocols, datasets, etc. relevant to these studies should be placed in appropriate separate subfolders in this module. Now, simplified trial summaries or ts.xpt files that are relevant to the method development, validation, and pivotal studies should also be submitted in Module 5312. For further details, please refer to Appendix C in the PDF of these slides. Information and data pertaining to analytical method validation or IVPT sample analyses belong in Module 5314. Thus, all relevant reports, protocols, SOPs, etc. relevant to analytical method validation or IVPT sample analyses should be placed in appropriate separate subfolders in Module 5314. And that brings us to our challenge question. Validated IVPT methodology as used in the pilot study should be utilized unchanged in the pivotal study. a change in which of the following would not be considered as a change in the IVPT methodology between the pilot and pivotal studies? A. Study procedure. B. Acceptance criteria. C. Formulations evaluated. Or D. Equipment. Take a few seconds to think about which answer you think is right. If you selected option C, you are correct. At slide 14, we identified options A, B, and D, but not C here as components of an IVPT methodology. Accordingly, use of a third additional formulation in the pilot study as compared to the test and reference standard formulations in the pivotal study is not considered as a methodology change. In summary, providing high-quality IVPT submissions is straightforward once we have a good grasp of their A, B, and C's. That is, a good understanding of the key components, the key submission expectations, and their organization within the submission. For comprehensive information, always consult the relevant agency guidances. Now, obviously, in order to generate high-quality IVPT submissions, the underlying IVPT information and data must also be high quality. Therefore, as you work on your future IVPT projects focused on high-quality developments and optimizations, validations, and pivotal evaluations, please utilize the A, B, and C's as discussed today to provide high-quality IVPT submissions to the agency. That concludes my presentation. I will be back shortly to answer your questions, and thank you for your attention.
Good morning, everyone. My name is Vipra Kundur, and I am a pharmacologist in the Division of Bioequivalence, Office of Bioequivalence within the OGD. Today I will be speaking on considerations for morphologically directed Raman spectroscopy, study information submitted in nasal suspension ANDAs. I would like to start my presentation with a disclaimer. The views and opinions expressed in this presentation are solely from myself and do not necessarily reflect the views of the FDA. The learning objectives are to explain the alternative approaches to the comparative clinical endpoint study for locally acting nasal suspensions, considerations for a high-quality submission for an MDRS study, and to identify submission-related common deficiencies for an MDRS study. The FDA has a weight of evidence approach for demonstrating bioequivalence for the nasal spray suspension drug products. The weight of evidence approach utilizes in vitro studies to demonstrate equivalent in vitro performance, pharmacokinetic or PK studies to demonstrate systemic exposure, and comparative clinical endpoint study to further support the equivalent local delivery for a test product that has demonstrated formulation and device similarity. However, there are certain challenges in conducting a comparative clinical endpoint study, such as high variability, low sensitivity, and longer study duration. Therefore, FDA also recommends an alternative BE approach in lieu of a comparative clinical endpoint study. Historically, the FDA has recommended an in vivo comparative clinical endpoint study to ensure equivalent local delivery of drug between the test and reference products. However, with the emergence of advanced techniques such as morphology-directed Raman spectroscopy, the FDA currently recommends that the prospective applicants may submit drug particle size distribution data in lieu of an in vivo comparative clinical endpoint study to support bioequivalence of nasal suspension ANDAs. With the emergence of MDRS technique, there has been an increasing trend of nasal suspension product submissions that contains MDRS study. Many MDRS study deficiencies are related to incomplete submissions. So in this presentation, I will provide an overview of common deficiencies that are observed for an MDRS study, and I will also provide clarity on the agency's expectations on MDRS data in support of bioequivalence. In this slide, I am going to talk about what information should be submitted for an MDRS study and also in which module should the information be submitted. All the BE summary tables should be submitted in module 2.7.1. Currently, we don't have BE summary tables posted for an MDRS study. However, we recommend that you please check the fda.gov website for the most current recommendations. In module 5.3.1, please submit the method development and method validation reports, including standard operating procedures and protocols with predefined acceptance criteria that were effective at the time of the study. In the same module, please also submit the pivotal study report. For the pivotal study, please submit the 100% raw data and also the PBE analysis on D50 and SPAN. Depending on the data submitted and upon evaluation of the said data, additional information may be requested during the ANDA assessment. In the following slides, I will be discussing the most common submission-related deficiencies for an MDRS study. These submission deficiencies are related to method development, method validation, and pivotal study. The most common submission-related deficiencies for method development are observed for sample preparation and image and size characterization. Submission-related deficiencies for sample preparation are mostly related to incomplete submission of detailed procedure and validation data. Therefore, we recommend providing complete optimization data on the following. Selection of an appropriate dispersion technique. For example, wet dispersion or wet measurement technique should be used for aqueous-based suspensions. We recommend providing particle size distribution data for pre and post actuation to demonstrate control on agglomeration. And also each batch should be evaluated separately. Data should be provided to ensure that the selected sample volume is appropriate and also has no touching particles and the dispersion is uniform. Particle size distribution data for each concentration should be provided to ensure reproducibility. We recommend providing optimization of particle settling time and also the method used for de-agglomeration. If any surfactant is used, then the solubility of particles in the selected surfactant should be provided, and also the concentration of surfactant should be optimized for particle size distribution reproducibility. Since excessive pressure results in particle damage, the cover slip and its placement is very important. Therefore, particle integrity validation data should be provided. Validation data should also be provided for the selection of an appropriate sealant for preventing sample evaporation. And finally, Data to justify the selection of number of sprays for sample preparation should also be provided. Image and size characterization deficiencies are also related to incomplete submission of validation data. We recommend providing supporting data for the selected thrash pixels and equivalent size for area pixel using appropriate magnification. Provide validation data to support selection of the threshold value. Optimization of Raman particle count data should also be provided. Validation data for distinguishing API and excipient particles and also agglomerates should be provided. We recommend that you specify whether the same elongation filter was used and optimized for both test and reference products. Provide data on selection of circular equivalent diameter that is appropriate for the product. Provide validation data to support the selection and optimization of Raman's correlation score. An appropriate orthogonal method should be selected to support the pivotal MDRS method. And finally, provide validation data to justify the pre-processing settings. Submission-related common deficiencies for MDRS method validation and pivotal study. Many of the deficiencies for method validation and pivotal study are related to missing study information and documents. We recommend that you submit validation data for MDRS method for reproducibility of the method and accuracy and also robustness of the method. For the pivotal study, please submit the 100% raw data and PBE analysis on D50 and SPAN. To summarize my presentation, the number of ANDA submissions containing an MDRS study has increased in recent years. I went over multiple common MDRS submission deficiencies that are related to method development, validation, and pivotal studies. And as you can see, many of the identified MDRS-related deficiencies could be prevented by providing supporting development and validation data. For folks who are interested in the CE credits, I have one challenge question for this topic. Which of the following statements is true? A. Potential challenges of a clinical endpoint study include high variability and longer study duration. Option B. Number of ANDA submissions containing an MDRS study has increased in the recent years. Many MDRS study deficiencies are related to incomplete submissions. Option D. All of the above. And the answer is option D, all of the above. So with this, I would like to conclude my presentation, and I hope with my talk, I gave you some helpful hints on how to improve your MDRS study submissions by avoiding unnecessary deficiencies and also reducing the number of review cycles. Thank you.
Hello, I am Scott Gordon. I'm a Senior Health Informatics Officer as part of the Data Standards staff in the Office of Strategic Programs at the Center for Drug Evaluation and Research at the FDA. I'll be continuing to talk about the topic of this session, specifically focusing on our efforts to standardize the information of pharmaceutical quality, chemical manufacturing and controls, or QCMC, specifically at a high level, what is it, where are things now, and how you can learn more. Standard disclaimer, the views and opinions in this talk are mine and shouldn't be attributed to my employer, FDA. And the presentation itself should not be considered guidance or a change in guidance, nor does it necessarily reflect FDA's positions. After this talk, you should be able to explain the problems the PQCMC program intends to solve and how, identify the goals, objectives, and scope of PQCMC, describe the processes used to develop the PQCMC data standard, understand stakeholder outreach taken by PQCMC, discuss some current and future work, and recognize where you can find out more. So why is PQCMC needed? What is the problem that the PQCMC program is attempting to address? On the left-hand side is a representation of the Electronic Common Technical Document, the ECTD, which is an ICH standard that is used for packaging all applications to regulatory agencies such as the FDA. For the purposes of this talk, focuses on Module 3. Module 3 is the home of all the detailed information relating to the product quality, chemical manufacturing, and controls for the drug product in question in a given application. Some information, relevant information, also exists in Module 2 in summary form. The issue at hand is that all of Module 3 arrives to FDA as essentially electronic paper, so PDFs and things of that nature. These are inherently unstructured and non-computable data, information. They're often not standardized. You can represent similar things in slightly different ways. For the sponsors, this creates a large amount of manual work just to create. And on the FDA side, there's an equally large amount of manual work to transcribe and otherwise prepare this information so that they can reliably analyze this information. The net result of this is it's incredibly time-consuming. Not an ideal way to be preparing data or using data that needs the kind of analysis it deserves in the 21st century. So what is the solution that the PQCMC program is offering to help address this issue? The PQCMC program is developing a data standard that supports the transmission and receipt and processing of PQCMC information that is consistent in format, uses consistent values, and most importantly is highly computable and ready for analysis. There's benefits across the board, and these are not exhaustive. From the sponsor side, having this as a standard in the future will allow some clear expectations of exactly how data should be formatted, will actually support a pre-checking of the content and the quality of the content before submission, and even support other industry-side data analytics. From the FDA side, we see this as being beneficial in providing a consistent in format and values that are received. It will be able to fuel software-powered analysis and will be much faster to review. And on the FDA side, we can also check for valid content and data quality upon receipt. As a mutual benefit, this is an example of the paradigm of submitting the single kind of information, the single piece of information one time to FDA, and FDA can then use it for many purposes, as opposed to sponsors having to submit basically the same information to different places at different times. What are the core activities of the PQCMC program? We'll start with the two overarching goals. First is to determine just how to structure Module 3 information, and then to work with a standards development organization, in this case HL7, to make this an implementable data exchange standard. And the PQCMC data standard is being developed using HL7's Fire data standard, and I'll be talking a little more about that in a moment. And as for the scope of the PQCMC program, the amount of information that this developing standard is meant to cover, it's quite a lot. It's pretty much everything that's amenable to structuring in Module 3, and as mentioned before, also some sections of 2.3. And it covers quite a lot of different types of applications, those that are pharmaceutical-oriented, those that are biologics-oriented, as well as veterinary medications. And the 3 centers listed here have been working since the beginning of the program on this effort as well. And it's also meant to cover pretty much all application types and pretty much all dosage forms. And now for a somewhat deeper dive into the information areas to be covered by the developing PQCMC data standard. The program divided this work into 2 somewhat sequential phases, the first of which covering many foundational concepts, and the second going deeper into complex manufacturing processes. So for phase 1, these are the foundational concepts. How to define every drug product and every substance within that product. The quality control tests and acceptance criteria for product substances, excipients, raw materials, formulas for making batches of the product, results of quality testing, stability, and analysis of batches of products and active pharmaceutical ingredients, as well as details on packaging and containers. And on the left, you'll see the more formal descriptions of these data domains. Phase 2 of this work then took all those foundational concepts and then dove into the description of the complex manufacturing processes for drug substances and drug products. So put very simply, how a manufacturer puts everything together to create products. Every step, every mechanism, machine, process, etc. And what steps take place at which facility, of which there are many for one product. And this phase is being done in a more iterative fashion. The drug substances, we are focusing first on small molecules, more of the pharmaceutical side, and then biologics. And on the product side, you can see we're aiming to cover a wide range of dose forms, the most common of which are things like solid orals, but there's many others, obviously liquids, and then getting into biologics and other variations. So the intent is to cover quite a bit. And also some additional coverage for things like annual lot distribution reporting and CMC changes in our annual reports. So a question I'm sometimes asked in regards to the PQCMC program is, why does it take so long to create a data standard? Well, creating a data standard from scratch is inherently immensely time-consuming process. And this is meant to be sort of an overview of why. When there is no pre-existing structuring, an enormous amount of time is spent on determining what can be structured and how to structure it. Issues such as the level of detail, number of data elements, terminologies that may need to be used, relative cardinalities of different elements, any business rules that FDA or another organization may have, all have to come into play for hundreds and hundreds of bits of information. All of this, of course, needs to be reviewed and revised. Internally, we review these at FDA with our various center subject matter experts. We've also, and I'll get into this more in a moment, released batches of these drafted elements and terminologies to public by Federal Register notice to get industry comment. And that also leads to revisions where deemed necessary. Then all of this is taken to the Standards Development Organization, HL7 in this case, to begin the sort of designing process. How do we represent all of this information, this structuring in HL7 Fire? And this is where we follow HL7 processes to the letter to ultimately create a final representation in HL7 Fire of the PQCMC data standard. The culmination of all this work at HL7 using Fire is in the production of what is called an implementation guide. An implementation guide, put very simply, is the set of instructions for IT folks to create and implement the software, databases, etc., to allow the creation of messages and or ingestion and processing of messages that follow the rules set down in the standard, all of this work that was developed. It also supports additional narratives and other information so that non-IT folks also will have as comprehensive an understanding as possible as to how this standard will work. As a brief aside, I'm sometimes asked, why use Fire for this work? Well, a number of reasons. First, of course, it's a consensus-based standard for public use. This is pretty much a must-have for U.S. federal government agencies. But more importantly, Fire has an immense amount of flexibility built into it and allows us to represent the data needs that we have quite exactly. There's a lot of work to make it do that, but once it's done, it works. Further, there's much built-in support in HL7 Fire for PQCMC definitions and concepts. Now, this is because of work that our teams at HL7, as well as the European Medical Agency and Health Canada did to put a lot of these requirements into what is about to be published, version R5 of Fire. So that's an extra benefit. Further, our PQCMC work harmonizes well with other Fire development in the same general data domain space. For example, the EMA work on IDMP. Now, I mentioned previously about the stage of review with industry, and this is just a blow-up sort of that previous timeline. The outreach to industry and the general public is critical to getting this right. Getting the input of all stakeholders is the only way to ensure that the ultimate representation of this information in the final standard will work for everybody. So the process that we at FDA take to getting this information out into the public is the release of what are called Federal Register notices, specifically asking for comment on documents. So we prepare documents that show a set of the data elements and relevant terminology lists and ask for comment. To be clear, this is not guidance. This is purely documents for comment. And once we receive those comments, our teams look them over, and often we get very useful advice, and that helps make refinements to the data elements and terminologies going forward. And here I'd just like to present a short timeline of the industry and public outreach that has been and will continue to be ongoing. So in 2017, we released a first draft of a good portion of the phase 1 data elements and control terminologies for comment. This was followed in 2018 by a public meeting, which gave us a lot of input about the whole PQCMC initiative, as well as additional feedback on phase 1. We had in 2019 a very early proof of concept IT testing with some industry participants that gave some valuable feedback at a very technical level. We spent the subsequent couple of years doing quite intense Fire development at HL7. So under the Biomedical Research and Regulatory Group, which is the sponsor of this work at HL7, and developing the Fire implementation guide. In March of 2022, we released a new notice showing an expansion of the phase 1 data elements with lots of revisions based on feedback by everything previous on this timeline, as well as a representation in a more document form of proposed representation in HL7 fire. And this is something we expect to be continuing now and into the future. So where are we now? Well, phase 1 structuring is complete, and phase 2 structuring is well underway at this point. An early draft of the HL7 fire implementation guide with phase 1 information is in development. This is a clickable link. Please go ahead and take a look. It is a draft implementation guide. It is information only as part of the HL7 PQCMC project. To be clear, it is not final. We expect revisions to it, nor is it binding in any way. It is purely for informational purposes. And revisions are expected as phase 2 structuring keeps moving forward and after public comment gets represented as fire in this implementation guide. Where are we heading? Well, we're certainly heading to the completion of phase 2 structuring. We'll be making decisions on staging, in other words, what parts of PQCMC to implement when. We definitely anticipate further rounds of industry participation in pilot testing. And as mentioned previously, we intend to continue outreach efforts and capture public comments in order to ensure we capture as much stakeholder input as possible to be reflected in the PQCMC standard. And finally, how can you learn more? We have a project page at fda.gov, and we plan to keep updating this periodically. We, as mentioned previously, are intending to have continued releases of more structured parts of PQCMC for public comment. There is a fire project page for PQCMC at hl7.org. This is a clickable link. And as mentioned previously, this is done under the auspices of the Biomedical Research and Regulatory Work Group at HL7. And finally, we do have a standing email address, pq-cmc@fda.hhs.gov, which you can use for any and all questions you may have about the PQCMC initiative. And that brings me to the end of my presentation. I'd like to thank you all very much for your attention.
Let's now turn our attention to the future of application submission and application review. After this presentation, you should be able to describe the PQCMC vision, explain what's been done so far, describe the role of these two standards organizations, explain our ongoing work and its challenges, and describe what you folks might do to prepare for the future of structured applications. So the current model could be said to be a narrative submission going to a narrative review or assessment from which data such as component and composition is manually entered into a FDA database. Then the PDF documents are stored in our electronic document room or the document repository. What might be called a future database model of submission would involve both structured Module 3 documents along with some unstructured narrative. For example, how it is that you arrived at the wet granulation model as compared to direct compression. The structured documents, as before, would be stored in our electronic document repository, but structured data elements such as components and composition would be automatically extracted into an FDA knowledge base for multiple uses, including to pre-populate CASA template review templates and to populate our facility risk ranking algorithm for inspection purposes. An important feature here is the 2-year publication timeline for when PQCMC would be a mandatory format. In this slide, I hope to clarify the distinction between PQCMC and CASA. Briefly stated, PQCMC is about standardizing submissions and CASA is about standardizing assessments with templates pre-populated from data that's retrieved from PQCMC submissions. So PQCMC's goal is to standardize and structure the quality information in the ECTD using data elements, data standards such as XML, JSON, and HL7 FHIR. CASA, on the other hand, is a system of internal knowledge management involving pre-population of review templates, risk ranking algorithms, and data analytics that will facilitate lifecycle management in comparison to previously submitted documents. In the end, implementation of PQCMC will enhance the current CASA system, which relies largely on manual population of the CASA template. So the important takeaway message is that PQCMC is about submission and CASA is about assessments. Here's a summary of where we are, having assembled a diverse range of subject matter experts across the 3 centers. We've created data models for the listed elements and standardized terms and definitions, relying first on existing standards, for example, from FDA, ICH, IUPAC, USP, and EMA. We have had a proof of concept with 5 pharma companies where we tested the FHIR instance could be successfully sent through our electronic gateway and that it did adequately represent the data prior to its transformation. We have worked diligently to harmonize with the CASA group, although initially we worked separately. We're also collaborating with internal FDA standards groups, which I'll mention subsequently. So what we've published is 2 Federal Register notices in 2017, which laid out our Phase 1 terms and definitions. The 2 main comments were related to expanding PQCMC beyond FDA efforts, which are described on the next slide, and conforming to the ISO identification of medicinal products standards, for which we created a document with a term-by-term mapping. And we also held a Zoom with pharma to engage in an interactive discussion. Later in my talk, I'll discuss some of these challenges with IDMP. And here are some references to IDMP and to our Federal Register notice, and a reference to our FDA webpage that contains PQCMC information. Along with Health Canada, we proposed an ICH topic on structured data quality. This proposal was accepted as a new topic called Structured Product Quality Submission to start after progress is made on the CTD-Q revision by the M4Q working group, which is ably chaired by Lawrence Yu. We anticipate next year as the start date for the SPQS group. But we'll continue with our efforts in PQCMC because we need them to support the CASA initiative. The history of the IDMP standards is that they were spun out of ICH pharmacovigilance groups in the early 2000s. Subsequently, the scope has expanded to cover exchange of all medicinal product data, for example, by creating a global unique identifier for a medicinal product known as PHPID. The 5 standards listed cover substance, dosage form, routes of administration, units of measure, and other medicinal product information. What conformance to IDMP means is not so straightforward. Does it mean having the same data model? Can there be the same data model with different terms? The EU, for example, mandates the use of IDMP, and yet the EMA used different term lists for ingredient roles. An effort to map dosage forms with EDQM resulted in only a 30% overlap of terms, and medicinal product ID is left to a regional implementation, and no other country, for example, uses our NDC code. And FDA requires USP dosage form labeling, for example, and even within FDA, we have several different dosage form lists that are in use. So the conclusion is that we'll try to align where possible with IDMP, but the conformance is just frequently not possible. The necessity for standard terms and definitions is to eliminate confusion about synonyms and the exact meaning of the same term used in different contexts. For example, until recently, the term active ingredient had different meanings within two sections of the Food, Drug, and Cosmetic Act. But standardization will not only facilitate an ontology or relationship between terms, but it will facilitate data analytics as given in the example below. It also will facilitate risk ranking. For example, does wet granulation give a better content uniformity than direct compression for low-dose drugs? So for us, we use these control vocabularies to create drop-down lists. For example, ingredient role would be comprised of these three items. To provide you with a specific example, here's what we did for drug product manufacture. By creating a model based on unit operations, what are those elements associated with the unit operation? They are category and subcategory, for which we had a SUPAC IR equipment addendum, which I'll mention was a great collaborative effort between FDA and industry, and for which there was a Hammer Award. So we also associated with the unit operation these elements like equipment data, process parameters, established conditions, in-process control, step ID, and material input. We had discussed adding established conditions, but because of the newness of this concept and the current work of the M4Q group, we decided to defer it and to not include it in the current PQCMC model. So here's an idea of what we do in our PQCMC collaboration with the CASA group, in which we look at common terms, different terms with the same meaning, and come up with a consensus on a given term, define it, and decide which group will make that change. Our current and future work involves these many external collaborators. Here and here, and many standards groups within FDA. We're pushing ahead into other sections. We have a JSON-based model for drug synthesis that renders a graphical synthetic scheme which builds in chemical intelligence and is not simply a picture. We focused so far on solid oral dosage forms, but we're now expanding into liquids, including biotech products such as monoclonals, with input from our Office of Biotech Product subject matter experts. And finally, late next year, we plan to publish a PQCMC draft guidance that will likely focus on specification, components, and composition. There are IDMP challenges in the road ahead, including its initial focus on pharmacovigilance, a different specification model, and use case, many undefined terms, and I would note that our contractors, however, required us to come up with a definition for every term we used, and every regional control vocabulary list. We had to have terms for each of those. And then there are many PQCMC related terms that are not in IDMP, which really had no focus on drug manufacturing. So some of those are shown in this list. Other challenges we face include too many standards and too few standards. Too many in some of the listed examples, and too few as listed in the gaps. Developing these data models, definitions, and ontologies is time-consuming and resource-intensive process when done in a systematic way. Another issue is that we have yet to see any commercial vendors supporting FHIR for regulatory submissions, although there's a lot of FHIR support in the EHR, electronic health record arena, and in the clinical area. And of course, we need to build database structures to support the incoming submissions. So in conclusion, PQCMC will substantially change things. It will result in significant changes for FDA and applicants. There'll be a need for new business processes and infrastructure. And we hope that PQCMC will enable alignment and be embraced by IDMP. And as for the future, 745A mandates 2 years after the final guidance. And remember, we haven't published a draft guidance yet. And the Step 2, there's an uncertain time frame for any Step 2 document from SPQS. So because these changes will implement years in the future, there's still time for you to prepare. And about preparing, some suggestions that I have are to maintain current awareness of these efforts through things like Google Alerts, looking at YouTube, which has a lot of videos from FDA, EMA, and Unicom, which is a European project involving IDMP, and of course look to the FDA webpage. Other things you can do are apply the master data concept of a database with a single source of truth for a term. Group your data according to IDMP concepts. You might create a new field, administrable dose form, which is an IDMP concept, and just do data cleansing and curation. So here are a couple challenge questions. Which of the following statements is true? PQCMC is primarily about application assessment. CASA is primarily about application submission and their assessment. PQCMC and CASA are primarily about application submission and their assessments. Or PQCMC will provide the structured data needed for CASA assessments. So I'll give you about 5 seconds to think about this. And the answer is PQCMC will provide the structured data for the CASA assessments. Of course, PQCMC is not about application assessment. Okay, moving to our next challenge question. Which of the following is not true? PQCMC submission format will become mandatory in 2025. PQCMC attempts first to use existing data standards. PQCMC is now an FDA-only project. PQCMC has published 2 Federal Register notices. I'll give you a couple minutes to think about this. And the answer is PQCMC will become mandatory in 2025 is not correct because we only anticipate publishing a draft PQCMC guidance next year, and that will be followed sometime later with a final guidance, and the final guidance then will take 2 years. So thanks for your attention, and we'll handle some questions at the end of the session.
Good morning, everyone. My name is Lieutenant Commander Nimi Matthews. I'm a Regulatory Project Manager with the Division of Project Management in the Office of Generic Drugs. As a project manager, I review incoming submissions daily, and a significant part of that process is our triaging based on the information provided in the accompanying cover letters. Today, I would like to share with you all a few suggestions regarding the information to include with your cover letters. Our learning objectives today are the following: to understand the purpose of a cover letter and the cover letter attachment, to examine FDA-issued guidance as it pertains to both these documents, to evaluate pertinent information to include in the cover letter based on the submission, to discuss the resources available to help create an effective cover letter for the submission's purpose. At this time, I also wanted to point out that these recommendations and suggestions shared today are consistently being updated via new guidance and maps, so please refer to those for the most current information. To begin our discussion, I wanted to go over the purpose of cover letters. So what is the purpose of a cover letter? The cover letter provides us the initial snapshot into the submission. It helps provide a summary of contents, identify the purpose, such as a response to an IR, resubmission after an action. It highlights the key elements included in the submission. It provides applicable regulatory statements, such as the MMA verification. And lastly, it helps FDA route and manage these submissions effectively. Similarly, let's define the purpose of a cover letter attachment. The cover letter attachment serves as a guide to help prepare the cover letter and submission. So it's very different than the actual cover letter. It serves as a tool to help prepare the cover letter and the submission. It is designed as a checklist for the common types of information applicants include in their cover letter. The goal is that this checklist format will help ensure that all relevant information outlined in the checklist is addressed in the corresponding cover letter. As mentioned earlier, this tool also helps FDA triage and manage the submission without unexpected delays. A sample of this checklist is provided in the draft guidance titled Cover Letter Attachments for Controlled Correspondence and ANDA Submissions, a link to which is provided at the end of this presentation. The use of this tool or its submission to FDA is not required, but it is recommended to help create a complete and accurate cover letter. Next, I want to share a few guidance for industry available on this topic. Our first one is the one for ANDA submission, content, and format, which was finalized in June 2019. As the name suggests, this document contains non-binding recommendations of the contents and format of an ANDA submission. As an applicant, you can always use an alternative approach to what is outlined in this guidance, as long as it satisfies all the requirements of the applicable statutes and regulations. What is really helpful in this guidance is the suggested cover letter template included in the appendix of this guidance, as well as the recommendations it provides on what should be generally included in the cover letter. On page 4 of this guidance, it provides some information about the cover letters, which is included in Module 1, with the other administrative information of an electronic common technical document submission. It also provides a non-exhaustive list of some of the major changes to an application that should be included in the header of the cover letter in order to avoid any triaging delays. Some of these include new strength amendments, changes to a dosage form, etc. The appendix I mentioned earlier provides a template for the cover letter. Not all the paragraphs recommended in this template need to be included in your cover letter. Only pick the applicable ones for that specific submission. It is highly recommended that you adapt the cover letter to meet your needs and submission type. You could also create your own template from the provided template and modify it for each of your submissions. The next guidance I wanted to share today is the one titled Cover Letter Attachments for Control Correspondence and ANDA Submissions. The draft version of this guidance was released December 2021. Appendix 2 in this guidance provides a cover letter attachment template, which is, as with the other templates, it is highly recommended that you modify the template to meet your submission needs. Of note, the cover letter attachment does not replace the cover letter itself and should be used as an add-on or a guide to create the perfect cover letter. The last guidance I wanted to bring your attention to today is the one titled ANDA Submissions, Amendments to Abbreviated New Drug Applications under GDUFA, which was finalized in July 2018. On page 15 of this guidance, it includes useful information regarding the contents to include in the cover letter, which we will cover in further detail in the next few slides. As with the other guidance, use the template and the information provided in the guidance and tailor it to meet your needs and your submission needs. Additional information to include in the cover letter can also be found in the specific guidance for that submission type or a specific map. FDA also has a quick access page listed here on this slide for all the available guidance and sections of the FDA maps that you may need to refer to. Our next topic of discussion are the components of a cover letter. The points I discuss on the next few slides are recommended for all submission types. First one is it is recommended that the cover letter be issued on the company letterhead. It should indicate the type of submission, such as a CR response, an IR, or a DRL response. It should include the date of the submission and reference at a minimum the ANDA number, the name and strength of the drug product, as well as the sequence number of the submission. Continuing on with our recommendations for all cover letters, the cover letters should additionally include a statement on how the documents were submitted and the file structure, the name, signature, and contact information of the person submitting this information, a regulatory and technical point of contact for the submission, including an email address, and reference, if any, to a relevant FDA action letters, emails, or correspondence. Of note, if the cover letter is signed by someone other than the responsible official or the U.S. agent, it should still include the name and contact person listed as the U.S. agent or the responsible official on the 356H form. Continuing on with our recommendation for all submissions, the cover letters should include a regulatory description of the submission, including an appropriate regulatory information and any desired hyperlinks to the submitted information. It should include a MMA or verification statement. It should include a technical description of the submission, including the approximate size of the submission, as well as a statement stating that the submission is virus-free with a description of the software used to check the files for that virus. Moving on to recommendations based on the submission type, please always refer to the applicable guidance for that specific submission type. Additionally, some submissions, like withdrawal requests, require that you reference the aspect of the CFR that is the basis of the submission in the cover letter. Provided here are two examples of how to use the header and references on a cover letter. Note how all the pertinent information is provided upfront, which is tremendously helpful during the triaging process. Now that we have covered a lot of the information regarding what needs to be included in the cover letter, this is a good time to pivot and share some of our frequently observed errors or omissions. First up is missing the MMA verification statement as required in 21 CFR 314.96D1, which requires amendments to an ANDA to contain the appropriate patent certification, recertification, or statement if the amendment contains new indication, new strength, changes to product formulation, or a change in the physical form or structure of the active ingredient. If the amendment does not contain any of these, the applicant should include a verification statement noting that the amendment does not include the changes I just mentioned. But one or the other is required for most types of amendments. To avoid delays and to err on the side of caution, I recommend adding that verification statement to every amendment. Next one is missing the priority request on every resubmission, even after it was granted. Resubmissions open up new projects in our database, and noting that a priority was requested and granted on the cover letter helps project managers ensure that the correct goal dates are assigned. Sometimes project managers assigned to the ANDA change, and this information upfront on the cover letter help avoid delays and errors during triaging. Next, highlighting any unsolicited information or major amendments in the cover letter upfront allows FDA to accurately assign a goal date upon the receipt of the amendment and reduces the later changes in classification when an unsolicited information or major information is discovered during the review of the submission. Some examples of this include gratuitous labeling information that is necessary for an approval. Next up on our list is not noting new or revised patent certifications, litigations, or carve-out updates. An additional point here is when including carve-outs in a cover letter, I recommend bolding them, having them in bolded font so they are easily identifiable and addressed accordingly. Following that is tucking in major changes or significant information on latter pages of the cover letter versus having them up front and on the first page, such as including a new strength amendment on page 10 of a lengthy cover letter versus having it right up front in the header of the cover letter where it's easily identified. And last on our list is not noting major amendment information upfront. A few examples of major amendments are new batch studies in response to deficiencies, changes in manufacturing sites. So the changes are made on the 356H or in the quality section, but it's not noted on the cover letter for us to triage that amendment appropriately. Any changes to a DMF or changes that would require an additional filing review, like a new strength amendment, but it's not noted on the cover letter. Instead, it's just noted in the body of the actual submission. For a full list of major amendments that are considered major, a full list is available in the guidance and the submissions amendments to abbreviated new drug applications under GDUFA, and it's in Appendix B of that guidance. Please refer to that guidance, and if any of your submissions fall under one of those categories, please remember to note and tag that cover letter with a major amendment notification clearly identified in the header. Now that we've covered some of the most common errors, let's move on to best practices. First up on our list of best practices is to include all new or major changes, including any labeling updates, right in the header or reference if you are combining submissions. Oftentimes, labeling updates are included in submissions along with other information, but not identified on the cover letter, and this can lead to delays in labeling information being reviewed. Always remember this. If the information submitted can in any way change that goal date, please put that on the header or reference section to be on the safe side. We highly recommend you create your own cover letter and cover letter attachment template to include all the information typically included in your submissions. It does two things for you. It reduces chance of leaving out any pertinent information required for most submissions, such as an MMA or a verification statement. It also helps you take out the inapplicable information and tailor that template or that cover letter for that specific submission. Next up is highlight any significant elements of your submission right at the beginning of your cover letter, as well as order your changes or order your submission from major to minor changes. So all the major changes noted up front and not lost in the latter pages of cover letter, as we mentioned earlier. Always remember this. If the information that is included can change the goal date, please put it in the heading or the reference section of your cover letter. Highlighting any unsolicited information or major amendments in that cover letter up front allows FDA to accurately assign the goal dates upon the receipt of that amendment, and it reduces the later changes in classification where we identify unsolicited information or major information during the course of the review. Continuing on with our best practices, please prominently identify or bold in your cover letter if a labeling carve-out is part of that submission. Separate each item in a paragraph of its own. Be concise, especially if more detail is provided within other modules. And use keywords rather than a vague description of the content. For example, using the keyword reformulation versus stating changes to composition of the product. Get familiar with the types of changes or information that can affect your review process and ensure that those changes are always noted on your cover letter body and header. For example, having keywords such as a new strength amendment or clearly stating that one of your facilities is not ready for inspection helps us triage that amendment accordingly. Bold the text of any administrative requests that are combined in with the data submitted for review. For example, if there is a request for reconsideration submitted along with the CR letter, please bold those so those are easily identifiable to the reviewers as well as the project managers. Use a cover letter and a cover letter attachment when combining submission types, so you don't have a 3-page cover letter, and if the project manager reviews the cover letter attachment, you can easily see all the different types of submissions that are combined into that one amendment. This is new for GDUFA III. So when requesting a change in CR classification for facilities only CR, using the correct keyword is just as important. So for facilities major to minor requests, include the keyword facility only or facility-based reclassification. We recommend putting this in the header as well as the body of the letter, and you can do that with the other recommendations I've mentioned earlier as well. Put those keywords in the header as well as in the body. And this new process is applicable only to CR responses and should be included with the CR response to be considered. Now that we've come to the end of our presentation, let's do a quick knowledge check. Challenge question 1. Which module of the ECTD submission is the cover letter contained in? Module B, Module 2, Module A, or Module 1? And the right answer is Module 1. The next challenge question is a true or false statement. The FDA cover letter template provides information that is required for each submission. A, true, B, false. And the answer is false. This is because the FDA provides a general recommendation of everything that needs to be included in a cover letter, but it is up to the applicant to modify that template and to adapt it to meet your needs and to only include pertinent information to that specific submission. Our challenge question number 3 is another true or false question. The FDA cover letter attachment can be used instead of the cover letter for the submissions. True or false? And the answer is false. As stated before, the cover letter attachment does not replace the need for a cover letter to accompany each submission. In summary, the applicant should use the cover letters and the cover letter attachment to help FDA identify the content and purpose of the submission. While FDA provides some guidance for cover letter and the attachment, the applicant should tailor their cover letter to meet their specific needs and the regulatory requirements. The cover letter should help guide the FDA on how to route your submission for an appropriate review. The cover letter should clearly state any significant changes to the application, such as formulation change or new strength amendments, right in the heading and the body of the cover letter. Cover letter attachment is an excellent tool to ensure all pertinent information is addressed in the cover letter and the submission. I wanted to share a few of our resources today with you, and these links listed on this slide will take you to our landing page for guidances, maps, the ECTD guides, as well as the CFRs. And listed here are the direct links to the guidances we talked about today. And as always, if you have any questions regarding the cover letters or the templates during the course of your submission, please do not hesitate to reach out to the regulatory project manager assigned to your ANDA. That brings us to the end of my presentation. Thank you all for your time and attendance today. I hope the information I shared with you is helpful and useful to you.
Thanks so much for all the great presentations. We'll now move into the Q&A panel session. And please remember, if you haven't had a chance to turn your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. And as another reminder, please remember to indicate at the beginning of your question the presenter that you wish to respond to your question. We'd like to welcome back to the Q&A panel Lieutenant Commander Malik Imam, who's the Deputy Director of the Office of Regulatory Operations within OGD, and Commander Andrew Fine, who's a Senior Advisor in OGD's Office of Safety and Clinical Evaluation, Division of Clinical Review. Looks like we have some questions coming in right now, and the first questions will be addressed to Commander Mahavidhan. And here's the first question for Commander Mahavidhan. Can we submit other related documents and the DRL response impacted due to the query, along with documents requested by the agency, in order to avoid further queries related to the same?
Thank you so much for that question.
In general, please respond with the requested information only, which will include any related documents that may help answer the DRL deficiencies. Note that if you submit any information that's not requested by the agency, the goal date may be extended. Thank you.
Thank you for responding to that question. We do have another question for Commander, and here is the question. What if I have a question for the agency and if it fits in multiple submission categories like a controlled correspondence or meeting, how do I know which submission path to choose?
Thank you. That's a really great question. So if you're unsure of the submission category, it's recommended to select what you think is the best pathway. If the agency determines that a different pathway is more suitable, we will suggest that to the applicant rather than just denying the request. And as a reminder, please do not submit the same question using multiple submission pathways. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a couple of questions that came in from Captain Keister. And this is a long one, so I can certainly read it if we need to. And here we go. If an establishment has been submitted in sequence as 0, 0, 0, 0, it is assumed that the answer to the question would be yes for is the establishment new to the application, and pending for what is the status of the establishment. But after the original sequence, if an amendment to an IR is filed as sequence 0002, still an unapproved ANDA, and if there are no changes to the establishments in the application, how is the circled question to be answered in this sequence? That is, for the question, is the establishment new to the application? Should the question be answered no? And the second question, what is the status of the establishment, remain as pending? Captain Keister, we're having problems hearing you. We're going to move on to the next panelist and let our tech team reach out to you to work with the audio. Moving on to our next panelist, we do have a few questions that came in for Hui Zhang. And here is the first question. Is it acceptable to use the test product in IVRT method validation?
Thank you for the question. Yes, it is acceptable. During the method validation, both the test and the reference products can be used. In addition, the applicants need to manufacture an altered formulation to use it to demonstrate the supplemental selectivity of the IVRT method. Thank you.
Thank you for responding to that question. We do have one more question for you, Hui Zhang, and here is the question. Should the applicant submit separate 2.7 tables for method development, validation, and pivotal study in ANDA?
Thank you for the question. Yes, please submit separate summary tables for the method validation, development, and the pivotal studies. Thank you.
Moving on to our next panelist, we've got a couple questions that came in for our Chana Manarika. And here is the first question. Would you please share the general experimental conditions that can be used to conduct IVPT robustness, as the general guidance does not include this information?
Yes, thank you for the question. As discussed in the IVPT general guidance, inherent variability in the permeability of human skin, whether in vivo or in vitro, is incompatible with the primary assumption related to robustness testing, which is that the test system performs consistently when all system variables such as temperature, stirring rate, etc. are at nominal settings. Therefore, while recommendations for alterations to study conditions for the purpose of demonstrating method robustness can be provided for IVRT studies, the same cannot be provided for IVPT. Nevertheless, over the course of your IVPT method development, if you note that the permeation profiles or IVPT endpoints are robust to certain variations in method parameters, such as, for example, stirring rate or the applied dose amount, then complete details of those findings should be reported in support of method robustness. Thank you.
Thank you for responding to that question. We do have another question that came in for Chana Manarikar. And here is the question. Can you clarify what evidence should be submitted to support consistency of study procedures and conditions and control method of parameters across the method validation and pivotal studies?
Thank you. This is a great question. As noted during my presentation, scientifically justified and optimized IVPT methodology, including suitable method parameters, study conditions, and study procedures that ensure precise control of method parameters, should be established during method development. Thereafter, these well-controlled study procedures should be meticulously documented in SOPs for use in the subsequent validation and pivotal studies. Accordingly, across these validation and pivotal studies, IVPT method parameters and environmental conditions, such as those described in Appendix B in my presentation, they should be monitored over the IVPT study duration, and evidence demonstrating their control within adequately justified and pre-specified tolerances should be submitted. Now, adequate control of method parameters over the study duration across each cell and each IVPT experimental run can be achieved when the corresponding study procedures are also similarly well-controlled and consistent. Thus, demonstrated control of IVPT method parameters along with detailed descriptions of how each study procedure will be performed and controlled can support a conclusion that the corresponding study procedures are also well-controlled and consistent. So to give you an example, relevant to validation and pivotal studies, evidence of adequate precision and accuracy of the applied dose, the actual applied dose, which is basically dispensed dosed minus any dose that is lost during the application process, along with granular description of the dosing procedure and how it is controlled in the relevant SOPs, together can be supportive of the consistency and control of the corresponding procedure, which is the dosing procedure. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we've got a couple of questions that came in for Vipra Kundur. And here is the first question. Do you know when the VE summary tables for the MDRS study will be posted?
Thank you for the question. Currently, we are working on developing the BE summary tables for the MDRS study. However, we don't have a timeline for when they will be finalized. So please note that the tables, when finalized, will be posted on the fda.gov website. Therefore, we strongly encourage you to check the FDA website for the most current recommendations. Thank you.
Thank you for responding to that question. We do have one more question for Vipra Kundur, and here is the question. Can you elaborate on what validation information should be submitted for the selection of circular equivalent diameter data?
Thank you for the question. For the CE diameter, we recommend that you provide validation data to justify the use of CE diameter that is not blind to significant % of the API particles. We also suggest to provide specificity, accuracy, precision, and robustness of the API particles using the selected CE diameter. Thank you.
Thank you for responding to those questions. We're going to check back in with Captain Keister and see if the audio works out. And here's the question that we previously asked, but we're going to ask it again since it's so long. If an establishment has been submitted in sequence 0000, is it assumed that the answer to the question would be yes for is the establishment new to the application, and pending for what is the status of the establishment? But after the original sequence, if an amendment to an IR is filed as sequence 0002, still an unapproved ANDA, and if there are no changes to the establishments in the application, how is the circled question to be answered in this sequence? That is, for the question, is the establishment new to the application? Should the question now be answered no? And the second question, what is the status of the establishment, remain as pending?
Thanks, Ray, and sorry to have to make you read that one twice. But in short, yes, what the questioner suggested would be correct. On initial submission, it would be new, and it would be pending. Assuming there are no other changes to that application, to that facility, rather than establishment, future submissions, you should not list it as new. That would, because it's previously been submitted to that application. And yes, it would still be a pending status.
Thank you for responding to that question. I heard you loud and clear, so we'll try one more question for you, Captain Keister. And here is the question. If I need clarification of deficiencies received in our CR, should I submit a post-CR scientific meeting request and a controlled correspondence?
Thanks for that question, Ray. For clarification questions, you can submit a request for a post-CRL clarification TCON or a controlled correspondence, but you should not submit both. If you also have scientific questions related to the equivalence, you can submit both these questions and a clarification question as part of a post-CRL scientific meeting.
Thank you for responding to that question. Moving down the line to our next panelist, we have a few questions that came in for Scott Gordon. And here's the first question. For HL7 FHIR, is the idea to eventually reduce the amount of PDFs submitted in Module 3 of applications? Dr. Gordon, we're having difficulty hearing audio. We're going to let you reach out to our tech team, let them check in with you, and then we'll check back in a few moments. We're going to move on to our next panelist, and we have a couple more questions that came in for Norman Schmuff, and here is the question. It's a little bit longer, so I'll try to read it slowly. I could understand how a description of a manufacturing process could be developed such that reviewers can have a complete understanding. However, that the document would not be like any existing master batch record. Is there a vision for how that regulatory document would relate to the master batch record that is on the manufacturing floor, driving the steps of the chemical operators and recording the relevant information in the executed batch record?
Yeah, it appears now that that information that we had structured in the manufacturing section would really have to be manually input into some application tool. In the future, for particularly as it relates to electronic master batch records and electronic batch records generally, we would anticipate that there would be an automated process where those items that would be submitted in the application, in the structured application, would be extracted in an automated way from the master batch record. But right now, and probably in the near future, it would require manual input and then an additional PDF file of the master batch record.
Thank you for responding to that question. We do have one more question for Dr. Schmuff, and here is the question. For PQ-CMC, whether the current ECTD publisher needs to modify their publishing tool, or is this needed for are any additional software tools needed?
Well, the initial the ECTD publishers have to be updated for version 4 of the new ECTD. But to get to this point of whether new ECTD tools will have to be available for structured submission, it may be that there is some back-end tool that's not actually part of the current ECTD ECTD tool that does the direct submission. For example, many of these files will just be, well, we anticipate they'll be XML and JSON files. So XML and JSON files presumably will be handled by any existing ECTD system that will handle either version 3 or the newer version of the ECTD system. So I don't anticipate that there'll be changes to that ECTD system per se, but you'll need some additional back-end tools.
Thank you for responding to that question. We're going to check back in with Scott Gordon to see if the mic and everything is working okay. And here's the question. I'm going to reread it. For HL7 FHIR, is the idea to eventually reduce the amount of PDFs submitted in Module 3 of applications?
The goal isn't to reduce PDFs per se, but to reduce to reduce the amount of unstructured, non-computable data coming in Module 3. So the goal really is to increase the amount of computable and structured data. So it's not about any one specific format, it's about whether it's computable and structured. Thank you.
Thank you for responding to that question, Dr. Gordon. We do have another question for you. We're able to hear you loud and clear. Here's the question. How can we learn more about the FHIR development of PQ-CMC?
That's actually a really good question. It is I think I mentioned, an official project at HL7. under the auspices of the Biomedical Research and Regulatory Work Group. In fact, I think if you go to slide 17 in my slide presentation, if you download that, there's a link to the PQCMC FIRE project page. This is the HL7 project. And that actually brings you to the Confluence page, which will give you information on how to join weekly, if you'd like, meetings of the Biomedical Research and Regulatory Work Group at HL7. Just a note, do look at the agendas. There's many projects at that work group. But on occasion, we'll be discussing things like PQCMC. It is a public forum, so obviously we would welcome all sorts of new participants. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We've got a couple of questions that came in for Commander Nimi Matthews, and here's the first question for Commander Matthews. Changes in dosage form. Did you mean to say changes in formulation? And the questioner provided a note. Usually a change in dosage form would require an approved suitability condition, a suitability petition, and a new ANDA.
Thank you for the question, Commander. And you are correct. I meant to say changes in formulation and not a change in dosage form. So thank you for the very attentive listener and the question.
Thank you for responding to that question. We do have another question for Commander Matthews, and here's the question. If labeling update is made due to changes requested in deficiency, is the labeling update considered unsolicited information?
So any new information outside of what was requested in the deficiency letter is considered unsolicited.
Thank you for responding to that question. We do have another question for Commander Matthews. It seems it'll be short, so here's the question. What is the MMA acronym meaning?
The MMA acronym stands for the Medicare Prescription Drug Improvement and Modernization Act of 2003.
Thank you for responding to that question. Moving back up to the beginning of our panel, I have another question that just came in for Commander Chitra Mahivathan, and here is the first question. Clarify in what instances is it more appropriate to submit a controlled correspondence versus a meeting request post-complete response letter, please?
Thank you so much for that question. So a controlled correspondence may be submitted for clarification questions or any questions that may be considered out of scope for a meeting. You can also submit a controlled correspondence after a teleconference if you're seeking further clarification or if you have any new or additional questions rather than requesting a meeting. And also post-complete response letter, you can submit a controlled correspondence to seek regulatory or scientific advice or to request evaluation of alternative BE approaches. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a question that just came in for Hui Zhang, and here is the question. For IVRT and a post-approval supplement, SUPAC-SS, do the same recommendations apply regarding information and reports to provide and placement in the ECTD structure?
Thank you for the question. The information presented today is applicable for IVRT studies with the purpose of establishing bioequivalence. If you submit IVRT data in a post-approval supplement for the purpose of drug product quality control, then the information should be submitted in Module 3. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a new question that just came in for Vipra Kundur. And here is the question. Can an in vitro study be an alternative to BE for nasal sprays?
Thank you for the question. For nasal sprays, the applicants may submit an alternative study proposal to demonstrate bioequivalence of their drug product. However, we strongly encourage that the applicants submit the specific alternative study plan to the agency prior to submitting an ANDA via appropriate pathways. Thank you.
Thank you for responding to that question. We have a couple more questions that came in for Chana Manarikar. And here is the first question. What information is expected to adequately support the qualification of receptor solution sampling?
Thank you so much for the question. As discussed in the presentation, the expectation here is that the suitability of the receptor solution sampling procedure is adequately qualified. So specifically, relevant to the IVPT validation and pivotal studies, adequate qualification or evidence should be supported in support of the following key aspects. So first is that the sample aliquot volume withdrawn at each sampling event is precise and accurate. 2, the sampling procedure is appropriate. And 3, the optimized IVPT test procedures ensure adequate sampling time precision from cell to cell within each experimental IVPT run. Thank you.
Well, that's all the time we have for questions in this session. A huge thank you to our presenters and panelists for the great presentations and answering numerous questions that came in. We'll now enter into our break for 10 minutes, returning at 11 a.m. Eastern Time. Enjoy your break.